So the first slide I will be finding is the letter E under sample slides. Now that I've had it put on the stage, I'm going to first do my coarse focus to be able to see it. And then I'm going to fine focus in order to make it sharper. So here is my letter E under our 4 power. I'm now going to switch to my 10 power. Fine focus a little more to keep it sharp. I'm able to move my slide around by clicking, dragging, and then unclicking. I'm now going to move to my 40 focus. I'm going to adjust my light a little bit. And now I'm going to move to my 100 power. Here I have to add some immersion oil in order to not crack the lens or the slide. Now I'm on the 100 power. Now that I'm done, I'm going to remove my slide by clicking Remove Slide. I'm then going to take my lens paper. This is Kim wipes. I need lens paper in order to clean the lens. I'm now going to find my plant cells under plant slides. Now that it's loaded onto the microscope, I'm going to first use my coarse focus. And then to clear it up a little bit, I'm going to use my fine focus. So this is the plant cells under my 4 power. I'm going to now move to 10 power. Only use my fine focus to clean it up a little bit. Move my slide around to see what we've got. Now I'm going to move to my 40 power. Use my fine focus to sharpen the image a little bit. Adjust my light. Now going to move to my 100 power, but again, I have to add immersion oil first. I'm going to clean up my focus again. Adjust my light. I'm going to move my slide around. So here, if you were to use the coarse focus, you will be going too far and you could potentially crack the lens and the slide itself. So now we're going to be very careful with our coarse focus. We're going to go back in with our fine focus to sharpen the image. I'm now done with my plant slide, and I'm going to remove that slide and clean our lens. I'm now going to move on to my spider. So you're going to go to Animal Slides from your catalog, and you're going to find spider leg. This is one option. You're going to use your coarse focus first. And then sharpen the image with your fine focus. I'm going to adjust my light a little bit. Now I can move on to my 10 power. All I need to do is adjust my fine focus, adjust my light a little bit, move my slide around, take a look. Now I'm going to move up to my 40. Move my slide around. I don't think I need to focus really. Maybe adjust my light. And then up to my 100. Add my immersion oil so I don't crack the slide or the lens. Adjust my fine focus a little bit. And my light. Now I'm going to remove my slide and clean with lens paper. I'm going to go back to the same catalog of animal cells, animal slides, and I'm going to look at the whole spider now. So I'm going to first coarse focus, and then sharpen the image with a fine focus. Adjust my light slightly. So we have a whole spider. Up to my 10. Fine focus to sharpen. Adjust my light to see better. Now we can look close at the spider. Up to 40. And all the way up to 100. So remember, you're only deciding on choosing one. Either use the whole spider or use just the spider leg slide. You do not have to do both. It is one or the other, whichever one you would prefer to look at. All right, I'm now going to remove my slide and clean the stage. And I'm going to move on to the bacteria slides in the slide catalog. And I'm going to find gram stain mix. All right, coarse focus first to bring everything into view. Fine focus to sharpen. Bunch of lovely blue dots. Move up to my 10. Now they look a little green, not blue. Now up to 40. Move the slide around. Now they look blue again. Then up to 100. Adjust our light, our fine focus. So now that we're zoomed in, we can see they are not actually blue or green at all. They are purple. blue and orangish red with the yellow. Very different. We adjust the light, we can see the colors a little better and the shapes. All right, I'm going to remove my slide and clean with my lens paper again. For this last one, you're going to be using the human slides. I'm not going to go through these because you can choose any one of them. This is one where you will have to do it on your own. Just make sure you let me know which tissue or slide you decided to use, and then give me your respective screenshots.